Good morning. I hope all of you are doing well. Now we're going to go over speciation. And if you will use your PowerPoint as a guideline, maybe take notes here and then look at your PowerPoint, that would be great. We know that islands provide especially good opportunities to study speciation, which is by definition the creation of a new species, and evolution. Macroevolution is the term that we use to describe the appearance and the extinction of species. But we have to remember macro, which means evolution on the large scale, occurs because of microevolution, which is evolution on a very small scale, which literally means the changes in the DNA sequences. Another thing that we have been working on in lab, and we'll talk about some in this particular chapter, is classification or taxonomy. We know that Carolus Linnaeus, devised what we call binomial nomenclature. Binomial means to name, nomenclature means naming. And it doesn't seem like such a big deal to us in this day and time, but you have to remember initially scientific names for living creatures might be 13, 15 words long. So this new characterization of species with only 2 names was pretty radical. He grouped creatures together that were physically similar, and he set up this very specific 2-name methodology, this genus for the first name, species for the second. He used a very specific hierarchical system to organize organisms. So what that means, this hierarchical system starts at the very top, as you already know, with domain. And once you have domain, which includes the greatest number of individuals, then you go from there to smaller and smaller classifications. So domain is the new one, okay? It's the largest group. Of course, Linnaeus would not have used domain. Domains are divided into kingdoms, and then phylums, class, order, family, genus, species, which is the smallest group. Now, there's been a lot of debate about how to really define what is, in fact, a species. Ernst Mayr developed what we call the biological species concept. Excuse me. And he said a species is a population, or maybe even a group of populations, whose members can interbreed successfully and then not only produce offspring, but those offspring have to be fertile themselves. He said that you can become an individual species as you accumulate differences in your genes. So reproduction with the other group is no longer possible. For example, we know that there are 2 different species of squirrels living on either side of the Grand Canyon, and they don't interbreed because of geographical separation for many, many, many years, and so they can no longer reproduce. There's some problems with his definition, though. First of all, it only dealt with sexually reproducing organisms. So people who were working with things like bacteria or fungi that reproduced asexually were like, hello, don't we get to classify our organisms? It also only dealt with living organisms. So people who were working in paleontology, working with fossils, were saying to Ernst Mayr, now how are we supposed to put our creatures into any category whenever they can't breed because they've been dead for millions of years? The other problem we have is some organisms of different species will interbreed if they are under the stresses of captivity, like in a zoo. They would not normally breed in the wild. And one that we'll be talking about a little later is like the liger, which is the combination of the lion and the tiger. And obviously they would not breed in the wild because they don't even live on the same continent. Some species also are not completely reproductively isolated. So it means that certain factors might keep them isolated, but then that might change. We'll talk about some examples of that. And they might, in fact, be able to interbreed. Okay. We're now on slide 6. I'd like for you to take a minute to look at all the different butterflies. And they look very similar to one another. How would you go about separating them into different species? By their colors? By their patterns? How many species do you count? And you know, we have to remember that even though they look alike, they can be quite different. And we'll talk about some examples of that a little later. We know that there are barriers to reproduction between different species, things that are designed to keep those species that are not the same from reproducing. And we're going to talk about some pre-zygotic barriers, and then we're going to talk about post-zygotic. Okay? So the pre-zygotic barriers are those designed to completely prevent fertilization. In other words, they are before the zygote has the opportunity to form. So we're looking at a zygote not being formed. And remember, zygote is the combination of a haploid egg and a haploid sperm to a diploid zygote. So what are some of those ways? Well, there's a great table in your PowerPoint. Have a look at that. The first one is ecological or habitat isolation. And this means, you know, you live in different environments. For example, if I live on the top of the mountain and you choose to live down near the lake, then we're probably not going to interbreed because we're living in different places. Or ladybugs. If I feed on a certain plant, like Mayflower, and you would rather be with morning glories, then we don't encounter one another because we have different eating preferences, so we won't be in the same place at the same time and able to breed. And that's what we call ecological isolation. Temporal isolation says you're active or fertile at different times. So because you are not ready to breed at the same time, then you won't. So for example, there is a really pretty flower called moonflower, and you can probably guess it blooms at night, versus morning glories, which open up in the morning. And because the pollen is not available for each of these at different times of the day, they will never interbreed. Behavioral isolation. This is when the organism has different activities like mating calls or perhaps dances, that kind of thing, that attract the members of their species but not of other species. Think, for example, how a cardinal might respond to a peacock spreading his tail feathers. He'll be looking at him like, you're kind of big and you look really stupid with your tail spread out. I'm just looking for somebody red. So we also have what is called mechanical isolation. And this is when the mating organs or the pollinators are incompatible. And so it literally means that because you have different physical structure, you cannot physically join with the other species. And this is particularly common in the pollinators like bees and butterflies, wasps, moths. Then there's gametic isolation. Gametic means gametes. You're isolated as a species because your gametes are so incompatible they simply will not join with one another. And this is true for different species of sea urchins, as an example. So we might be in the ocean and you would see the gametes, female and male, of different species of sea urchin be simultaneously released, but you don't get hybrid sea urchins because their gametes do not unite. So all of these go in the category of how do we go about preventing in nature the formation of zygotes. And they're ecological, temporal for time, behavioral, mechanical, and gametic. Okay. Excuse me. Now we're going to talk a little bit about post-zygotic barriers. And these are those where, yeah, we went ahead, the gametes joined, we form a zygote, but there's some kind of mechanism operating that prevents either viable, which means living, or fertile, which means can produce offspring themselves, offspring. So post-zygotic prevent viable or fertile offspring after the formation of the zygote. Hybrid inviability. In this case, hybrid means the joining of two species, but in means not, and viable means living. And what happens here is the offspring do not survive to maturity. So not only do they not live, they're not here on Earth long enough to reproduce. Hybrid infertility, which is the same thing as hybrid sterility, means your offspring are unable to produce because they are sterile. So a great example is a mule. If you want a new mule, you're going to have to go back and breed a horse and a donkey to get a mule, because mules are sterile. Same thing with ligers. Lion plus tiger gives you a liger, but it is a sterile offspring, cannot reproduce. Hybrid breakdown says the second generation hybrid offspring have reduced fitness of some sort. They're not going to survive. For example, if we breed hybrid mosquitoes of different species, the offspring have abnormal genitalia. That's their sex organs. So what a great way to not have to spray pesticides and still control mosquitoes. All we're doing is producing offspring that cannot then have sex and produce children themselves. Those are post-zygotic. Now, there are three spatial patterns that determine the three types of speciation. But I want you to remember there are a lot of gray areas. It's often very difficult to tell what type of speciation has created or maintains a certain species. Allopatric speciation is an example of speciation that has occurred because there's no contact between the populations. Usually, it is some sort of geographical barrier. Like, remember earlier I mentioned the squirrels that were isolated because of the Grand Canyon? These isolated populations, because they have no contact, completely stops gene flow and guarantees that the species will remain separate. This was also true, remember, of the Galapagos tortoises that lived on different islands. They all had their individualized, specialized adaptations, and because they lived on different islands with different amounts and sources of food, they developed different physical characteristics. Another type of speciation is parapatric speciation. And by the way, in your PowerPoint around slide 11 through 16, 17, 18, 19, there are good pictures of this, so please take a look at those. In parapatric speciations, the populations actually do share a border area. You know, like, so you might have something like one bird living where it's primarily grassland, another bird living where it is tropical rainforest. But where the grassland and tropical rainforest meet, there's going to be kind of an intermediate area. So they could breed if they went to that area. However, most of them do have a preference for habitat, and most of them mate within their own populations. Still, you may have limited gene flow. The organism may breed in the border area. Sympatric speciation, which is really wild, says there's continuous contact between the populations. There's plenty of opportunity for them to interbreed. It could be tiny little micro environments, you know, small spaces within a larger environment. Or an even better example is found in polyploidy in plants. For example, you remember that when we did the DNA isolation, we used modern genetically created 8N strawberries, which means that they had 8 copies of their chromosomes. Well, if you have a field of modern strawberries that are 8N growing, they cannot interbreed with the wild type, which only has 2 copies. Even though they're both strawberries, they're genetically incompatible, even if they're growing side by side. And remember, it can be difficult to determine the type of speciation and what barriers are important to keep the species separate from one another. We know, and there's lots of argument. Sometimes it seems like scientists really love to argue, but speciation may be gradual or it can occur in rapid bursts. Gradualism describes the gradual form where evolution occurs in tiny step-like changes, and it takes many, many generations, and it occurs very gradually, very slowly. Punctuated equilibrium says you get very brief bursts where you have rapid evolution, and then you get long periods where you have very little change at all. And it used to be that we were trying to argue, now which one of these theories is correct? Gradualism or punctuated equilibrium? And we now think that it's probably actually a combination of the two. Adaptive radiation, which means you have a species living in one specific area and then branches out to other areas like the tortoises on the Galapagos Islands, can give bursts of speciation simply because you've got a population inhabiting varied environments, and this gives rise to multiple specialized forms in a relatively short time. And there's a great example in the PowerPoint about the anole lizard in islands. This also can occur following mass extinctions where large numbers of creatures become extinct at the same time. Speaking of extinction, this is when a species ceases to exist. And typically what has happened, they cannot adapt to the environmental change that has occurred. What is most important is how fast are those environmental conditions changing? Smaller populations, for reasons that we'll talk about later, are at the highest risk of extinction. We know that extinction rates have varied over time. On Earth, there has always been what we call a background extinction rate. And that means that there's somewhere between 0.1, 1/10, to 10 extinctions that occur per year per million species. And that is if we as humans don't do anything to interfere or any other factor. Mass extinctions are whenever you get large numbers of creatures that disappear simultaneously. Please watch and take notes on the video and submit that to me by email the day the Mesozoic died. So some people believe in the impact theory that's described in that particular video, which talks about when meteors hit the earth. Some people also believe that you can get mass extinctions when you get dramatic movements of the crust of the earth. Now, one thing to remember is modern day classification systems are based on what we call common descent, where we believe that all organisms are related to a previous ancestor. This hierarchical methodology organizes species into groups, which of course begins with our 3 domains. A taxon is the term that we use to talk about a group at any rank. For example, class Mammalia, okay? Cladists, which are scientists, use cladograms to show the hypothesized evolutionary relationships of organisms. And toward the end of this PowerPoint, there are many examples. Phylogenies show the relationships based on evolution, and they use lots of lines of evidence, like fossils and DNA, embryology, structures. Cladistics defines groups by distinguishing between ancestral and derived characters or characteristics. Ancestral ones are inherited attributes that are still very much like the ancestor of the group. Derived are features that are different from those found in the group's ancestor and have been derived in this modern day form. And as you can tell from the slides, we can use these different characteristics to make a tree-like diagram. And we use the shared and derived characteristics. And we're trying to focus on who is related historically. Cladograms show the nested hierarchies of these relationships caused by evolution. And we can use many, many characteristics, including changes in DNA sequences. If we have enough information, we can even create what are called molecular clocks. And those can tell us, based on the number of changes, about how long, time-wise, it has been since the species have been closely related genetically.